right, good. So let's begin. So I'm going to give you a section on native mass spectrometry, and that you heard a little bit of that in Rachel Liu's talk when she talked about ionization methods. And so I'm going to expand on that topic first, and then I'm going to combine that with top-down mass spectrometry, because you'll see how some people in our field now are combining the two methods, and so it's easier if I just go from native MS and then next then to top-down mass spectrometry. Okay, so we're going to focus on mostly proteins now, and we've heard a little bit about that already in the earlier lectures. So mass spectrometry then is a premier method then to study proteins. The mass spectrometer it's a fancy balance then to measure large molecules like proteins, and from that mass measurement, what we get is sequence information. We get information on post-translational modifications, which some of you heard that earlier today, and molecular interactions, that's where the native mass spectrometry topic then comes into play. And so as you'll see then, as mass spectrometry has progressed, especially with native mass spectrometry, we get information beyond primary structure then. So you heard a little bit about that from Josh's talk about tandem mass spectrometry and proteomics. Now, we've now gone all the way to quaternary structures now to look at interactions between protein interactions, protein nucleic acid interactions, ligand interactions, all of these assemblies that are important in a lot of biological systems. So that's the strength then of native mass spectrometry. We can study these assemblies all at once. And so in Rachel Liu's talk, you heard a little bit about electrospray ionization, and the native mass spectrometry uses mostly electrospray ionization because not only does it not fragment molecules directly in the ionization event, it also allows other molecules that might interact non-covalently to a protein or to other molecules to be measured together. So if there's an interaction through hydrogen bonding, electrostatic interactions, if they have an interaction with another molecule, then you might be able to then preserve that assembly and then make that mass measurement. And so the beauty then of native mass spectrometry is these interactions can be preserved for mass spectrometry measurements. Our laboratory, many laboratories study ligand binding to protein targets, and so when a ligand binds to a protein, we can measure that size. We can determine from that size measurement then how many ligands are bound to a protein. That would be stoichiometry. And we can even determine solution phase affinities, like a KD, like in a biochemical sense, what the binding strength is in solution by this native mass spectrometry technique. Okay, so the raw data looks a little bit like what Rachel was showing. So when you have an electrospray spectrum of a large protein, you see all of these multiply charged peaks in a mass spectrum. And if you deconvolve that mass spectrum into the mass domain, you would get a molecular weight measurement then for that protein. So if you took a protein and you put it under what we would call denaturing conditions, meaning the type of solvents that you would use for HPLC, like acetylnitrile water with acid, that protein is going to be pretty much unraveled in solution. So we call that a denatured protein. And so you would measure then the mass of that denatured protein. This is an example we measured a long time ago. This was an enzyme that a group at Michigan had given us, and they wanted to know what the assembly size was under native solution conditions. So we took that protein, it was a dioxinase enzyme, and we took that protein and we dissolved it in a solvent that contained acetylnitrile, a little bit of formic acid, so the pH was low. Proteins unfolded, and we measured the molecular weight of about 44,700. That's the mass of the protein if you just calculated that from the polypeptide sequence. But then we took that same protein then, and we dissolved it in a solution containing just water with a little bit of ammonium acetate around pH 7 now. So that's the physiological pH of of most solutions. And we see then that the spectrum has changed, right? We see lower charging, fewer peaks, higher M over Z. Rachel talked a little bit about that when proteins are folded. And from the charge states and from the peaks then, it measures to be about 135 kilodaltons. So it went from 44.7 kilodaltons to 135 kilodaltons. And so it's jumped up by almost a factor of 3. So it's a homotrimer complex. So 3 subunits come together as an assembly. Plus there's a little bit of extra mass because this happens to be an iron-sulfur cluster. So there's these irons and sulfurs in excess that all conspire to be the full assembly of this enzyme. That's what the researchers at Michigan wanted to know at the time. And so that's just 2 measurements then to determine what's the assembly size, what are any cofactors that are present, what are any ligands present, just from this mass measurement. The classical kind of example that people would do when they first get into native mass spectrometry would take like some blood. You can take some blood, isolate the hemoglobin in it, and you should get a set of 2 peaks. These peaks here are the full tetramer assembly of hemoglobin. It has 2 alpha chains, 2 beta chains, and each chain binds to a heme molecule. It also has a natural stoichiometric subcomplex of a dimer composed of 1 alpha chain, 1 beta chain, and each chain binds to the heme molecule. So you see this natural equilibrium between the tetramer and the dimer. And you get that also then from this single mass measurement. Okay, so native mass spectrometry has has really progressed over the years as mass spectrometers get more sensitive. The mass range gets larger then, so that we can look at larger complexes. Native mass spectrometry as a term, though, was first coined in 2004 by Albert Heck. And if you read this paper that was published in JASMIS some time ago, he describes the sort of the genesis of the term, and he also reviews then what's possible with native mass spectrometry. But of course then, every time you coin a term or a field, then people come out of the woodwork and say, well, I was doing that before 2004. And so many of us were doing native mass spectrometry before 2004, before the term came out. And so let me just tell you about the genesis then of where this came from. So remember that John Fenn developed electrospray ionization in the late 80s, middle to late 80s, right? And so by 1990 then, people were investigating electrospray ionization for protein measurements. In 1990 then, this group at Rockefeller University, Brian Chait, had shown that if you took a simple protein like cytochrome C, just a single chain protein with a heme covalently bound, and if you electrospray that under different pH conditions, you get different charge distributions. You see that at low pH, you see this charge distribution peaking at 16 plus. If you go a little bit higher in pH, you see at pH 3, you see 2 distributions now centered at 2 different pH charge states. And then it goes slightly higher, then it collapses into 1 charge distribution. What's happening is that the protein is folded under different conformations, and under these pH conditions, the shape of the protein, the assembly is slightly different at the different pHs. The molecular weight's the same, but the charging is different. And so this was the first paper that kind of showed that solution conditions do affect protein folding, and that protein folding can be somewhat capitulated in the electrospray spectra. Okay, so then around the same time then, a group at Cornell, Bruce Ganem and Jack Henyon, then showed that now you can actually use electrospray ionization to measure a protein interacting with something else. Like in this paper, it was a non-covalent receptor ligand complex. This is FKBP binding to an FK506 ligand. So it was a ligand binding to a protein. This other paper that came in the next issue of JACS was an enzyme substrate complex. So they took in hen egg white lysozyme incubated with some saccharides, and you could see the enzyme lysozyme chewing up the saccharide, but pieces of the substrate were still bound to the enzyme, and so they could capture that in the spectra. So they can capture then interactions of small molecules to a larger protein and make the measurement then by native mass spectrometry before the term came out. Today now we go to much larger assemblies now. So native mass spectrometry has progressed now where people just measure larger and larger complexes. This is some measurements we made in our laboratory, all the way to 1.8 megadaltons in size for large protein-protein interactions assemblies. And now it's gotten kind of ridiculous now. Now you can take a whole virus, shove it into a mass spectrometer, and make the size and make the mass measurement of virus. So Albert Hecht's lab at Utrecht University, this is an early example. This was an HBV viral capsid assembly, one at 3 megadaltons in size and another one at 4 megadaltons in size. And the record that I think he's published, I think this is still the record, this is like almost 18 million molecular weight with a ridiculous elemental composition. You can see that, so in order to measure the mass relatively accurately, you still need to see charge states. You need to figure out what the charge state of these peaks are. You can see it under those conditions. You can see these little bumps, and they do line up to a charge state. So this middle peak would be 350 charges. The next peak over would be 351 charges, 352. And from those charge state assignments, then they calculated this to be this 18 megadalton molecular weight. It's not a routine measurement, I would say, and so it's not something that would be for the faint of heart. Not every mass spectrometer could do this, but still, it's kind of demonstrates what the size limitation of the measurement is, and it wouldn't surprise me if they've gone much larger than this, even as the instruments get better. This was in 2013, before some of these Orbitrap instruments that we're all using now were even developed. Okay, but so then, I think practically I think one of the biggest applications for native mass spectrometry is just measuring protein ligand interactions, like in a drug company, like in a pharmaceutical company. They might want to measure a small molecule drug binding to a protein target that the company is trying to develop inhibitors for. I used to work at Pfizer, and so we were doing this. We were doing these kind of native mass spectrometry measurements to take compounds that medicinal chemists would make. They would decide that this compound they think targeted this particular enzyme. We would take those sets of compounds, measure the binding of that molecule to the protein target. If the molecular weight shifts by the according mass of the drug, we can say, yeah, it binds. Or we can say 2 molecules of drug bind to the protein, not 1 molecule. So we can give that kind of feedback. In a drug company, then they would want to get the 3-dimensional structure of the ligand protein complex, and so we can tell the crystallographers, the NMR groups, then, yep, you should focus on this drug protein combination and spend all your time trying to solve the structure. So that's what we were doing back in the old days when I was working at Pfizer. We're also trying to make measurements of like ligand binding affinity, like solution binding constants. And so if you're a biochemist, you would titrate in some ligand into your protein target and make some sort of absorption measurement or some sort of affinity measurements. And then if you wanted to calculate a KD, then you would just titrate different amounts of compound. You would plot this in a scattered plot-like analysis, and from the slope, then you get a KD. It's the solution phase KD that you would report. And you can do this for compounds, that have affinities of micromolar, maybe sub-micromolar could be possible. But it's quite possible to use electrospray ionization and native mass spectrometry to make these measurements. Okay, so how many of you have done some of these measurements before? How many of you have tried to make a native mass spectrometry measurement? Anybody? Some of you have. Okay. So I want to focus a little bit on the experimental conditions, because it's not the routine way that you would inject a sample onto an HPLC and then get a mass spectrum. With native mass spectrometry, we're not quite at that level of automation, it's not quite just an inject a sample and you get a spectrum, although it can be, as I'll discuss in a second. Sample preparation is key. We heard a little bit of that in Bob Kennedy's talk about chromatography. Chromatography can't solve everything as far as sample prep, and the same is true with mass spectrometry. And so sample preparation is critical. How you introduce the sample is critical, especially for native mass spectrometry. There's a little note at the bottom of the slide. Online separation with native mass spectrometry is not routinely used. But size exclusion chromatography with mass spectrometry and even capillary electrophoresis with mass spectrometry are gaining popularity today, including with native mass spectrometry. So if you want to study complex sizes, ligand binding interactions, and you have a mixture of samples, SEC with mass spectrometry or CE with mass spectrometry is becoming a little bit more popular. It's still not widely used, but it's coming online. Okay, so let's talk about sample prep. I'm not going to go into great details. There's a paper that, if you want to do this experiment, there's a review paper that I'm part of a large consortium, and we published this paper in Natured Methods some time ago that goes through various protocols on that's based on the type of samples you might have, what might be in your samples. Is it a membrane protein? Is it a soluble protein? What kind of salts does it have? And it goes through different steps on how to remove that material so that you can get into the mass spectrometer for native mass spectrometry. So I'll just refer you to this particular paper. But some general comments then for native mass spectrometry. For most complexes you're going to want to study, you want to stay near physiological pH. So pH 7, 6.8, 7.2, something like that. Most proteins stay folded under completely aqueous solutions, so you're not going to be putting in acetonitrile or isopropanol, methanol into your solutions, even though people think it enhances ionization properties, but that's going to unfold your protein. So if your goal is to understand protein interactions with another molecule, you're going to want to stay away from those organic solvents as possible. Buffers, you want to buffer your samples to something close to pH 7. Ammonium acetate, not a buffer, but it is a salt that kind of keeps you close to pH 7. That's pretty much the only salt that people use in native mass spectrometry. Ammonium bicarb to some extent, but the pH is a little bit high. And people use around anywhere from 5 to 200 millimolar concentrations of ammonium acetate to keep the solution happy for native mass spectrometry. Some people can go up to 1 molar ammonium acetate if ionic strength is important. Our lab goes anywhere from 100 to 300 millimolar, just as a natural way to start the experiment. Other buffer salts may form non-specific adducts. So sodium is a killer. We'll see sodium adducts onto the protein. If it doesn't matter, you just want to know if a ligand binds to your protein, the sodium could be in there as long as it's not so high and as long as you can resolve all the adducts. But if you can't, then you want to get rid of the salt as much as possible. Rachel talked a little bit about the emitter sizes for electrospray ionization. You could do regular flow electrospray, so something like a microliter a minute up to 5 microliter a minute, and you can pump that in and still do pretty decent native mass spectrometry. It's not recommended. You're wasting a lot of sample when you're doing that, but if your system is just set up to do that, and you can make that measurement. For larger proteins, that'll become problematic because the droplet sizes are large, and it's tougher to desolvate a big droplet than a small droplet for native mass spectrometry. We generally use these nanospray tips that Rachel showed earlier yesterday. You can either buy them commercially, so Thermo sells them under the name Proxion, a company that they purchased. You can buy them from Waters, and there are some other companies that used to be able to buy them. I'm not sure if you can buy them anymore, but those are the 2 companies that I know right now that will sell them. But if you're making a lot of measurements for native mass spectrometry, then what people mostly have done is that they buy these pullers that will pull these capillaries, and they have different programs so you can make different dimensions of the capillaries, and then they would coat them either with a metal coating like gold or platinum, or put a metal electrode at the back end to get conductivity for the electric spray source. You can automate native mass spectrometry, although I don't see this very often now. This company called Advion used to sell a microchip that had these little nozzles at the back end. It had something like 100 nozzles per chip, and you could interface that with a pipette tip, and put in liquid in through each little bit of each nozzle to make an electrospray emitter for nano electrospray. But I haven't seen anybody use that in a long time. But if you have lots of samples to go through, this might be a way to go. And then Rachel showed this slide. I thought it was useful to show it again. The sizes of the emitters then do have an effect on its ability to get the sample into form for native mass spectrometry, and also has some ability to desalt by itself. The Williams laboratory at Berkeley, then, they showed that using a regular nanospray tip of about almost 2 microns in diameter as a tip, for samples that have a lot of salt and you haven't done anything to get rid of the salt, you can get a spectrum of your protein. This is a membrane protein, so it's got about some things like octoglucoside detergents in there. You can get a spectrum, but it's kind of ugly. But if you pull your tips to have very narrow diameters, then you get much nicer looking spectra, much nicer charge distribution measurements then. And some of the salt effects then is reduced. The salt is still on these peaks then, so there are still adducts here, but it's greatly reduced. I mean, this sample has 25 millimolar tris HCl plus 150 millimolar sodium chloride. There is still salt on these peaks, but at least you can get a mass measurement then of the protein complex this way. It's still better, though, to desalt properly. And so if you have the ability to desalt before you go into nano electrospray tip, then you're better off. Here is an example from our laboratory. This is a commercial sample, pyruvate kinase. It's a 232 kilodalton homotetramer. Without too much desalting, then you get a blob. But you get peaks here that tells you that something around 200 kilodaltons in size. But if you go through like an offline SEC purification protocol, you see that the peaks are much sharper. You get much more accurate molecular weight measurements then of the complex. So proper desalting can't be completely replaced, but kind of depends on what level of accuracy do you want in the measurements, what's needed for the experiments. And then I think lastly then for sample preparation, there is a method then that thermocells. It was developed by the Wysocki laboratory at Ohio State. If you buy this little short SEC column then, and you buy the proper injection valves, you can hook that up to your mass spec, and you can just inject your sample onto this little stubby SEC column then. It will desalt in addition to elute your protein directly onto the nanospray tip then, and you can get then a pretty fast mass spectrum then, native mass spectrum of a protein that might contain a lot of salts, but it's still under native conditions so that you can measure the size of these interactions then, of this interacting complex then. Okay, so I'm going to stop here for now. So I wanted to just see if you have any questions about what I've discussed so far. Sample preparation, methodologies, anything related to that for now. This slide just says that all kinds of mass spectrometers can be used for native mass spectrometry, including QTOFs, FTICR instruments, Orbitrap instruments now. And so all of these instruments that have sufficient mass to charge range, especially if you're trying to look for large molecules, you have to have enough resolving power so that you can see charge states for large complexes. All of these have been used, and so most instruments now can be used for native mass spectrometry. Any questions at this point then? Everyone know how to prepare a sample? Nobody wants to prepare a sample. All right, we'll get into this a little bit more. All right, so And so there is one challenge then, or many challenges, but one of the challenges with respect to different types of protein complexes are membrane proteins. Membrane proteins are tricky. They're not soluble in water. They're very hydrophobic, but drug companies want to study them because they represent important drug targets that really have been unexplored by pharmaceutical companies. But with the membrane protein then, you need to somehow solubilize them enough then for mass spectrometry analysis. And with membrane proteins, you have limited options, I would say. You have detergents to make a micelle. You have these other vehicles like nanodiscs and these other ones that are shown here that you can solubilize these proteins in these vehicles. They're going to be water soluble then at that point. But then when you put that into the mass spectrometer, you've got this crud with your protein, and nobody wants to make the measurement of the micelle plus the protein or the nanodisc plus the protein. You want to make the measurement of the protein itself. So somehow you have to be able to then put this into a mass spectrometer and then get the protein out of these vehicles so that we can make a measurement of the protein by itself without the vehicle. So several laboratories have figured out how to do that. It's all in the gas phase. So once the protein is dissolved, let's say this protein is in this micelle, it's in a water droplet. Somehow, as you electrospray that, you have to affect desolvation. So you have to get rid of the water in the interface of the mass spectrometer, the front part of the mass spectrometer. And then you have to pop out that protein out of that micelle. So you have to give it some more energy to pop that protein out so that you make the measurement of the membrane protein itself without the vehicle, without the micelle or the nanodisc. So there's some tuning going on. Every instrument does it a little bit differently, but once you've figured it out, you can get some quite nice looking spectra. So here's one from our laboratory. This is this protein called aquaporin. It's a water channel protein. It has 6 transmembrane domains per monomer. It sits on the cell membrane. Water comes in from the outside and goes in through one of the channels into the cell. That's the purpose of this aquaporin complex. And if you dissolve it in a detergent and then inject it into, say, an Orbitrap instrument, you can adjust the parameters such that the protein pops out of the micelle and you get a nice looking native mass spectrum of the protein. So this is a homotetramer complex, and you see a charge distribution that gives you the molecular weight of 97 kilodaltons, exactly what we expected then for this homotetramer. And then you can start looking at lipid interactions. So as I said, I have a slide on lipids. And you can actually take, under ideal conditions, you can take the intact membrane and then solubilize that in a detergent and then put that into a mass spectrometer. So here's an example we just published a year ago. This is a protein called bacteriorhodopsin. It's a protein with 7 transmembrane domains. It exists as a monomer normally. But when you get this protein in this particular microbial organism, it exists in the cell membrane called the purple membrane. It has a retinol that's bound to the protein, making the protein purple. And in this particular organism, the only membrane protein that's present on the cell membrane is this protein. So there's no other membrane proteins present for this microbial system. So it's kind of nice. You can just take the membrane and you've essentially purified the protein. But now you've got all the lipids that's sort of in the cell membrane. And so there's about 25 lipids that are present in the membrane of this particular organism. But these are the 2 lipids then that are thought to bind to this bacteriorhodopsin. Out of the 25, there's only 2 that bind to it. And so what we did was we just took a sample of the purple membrane, dissolved it in octoglucoside. So here's my student holding the solution of the purple membrane, so it's purple. And you just put this into the mass spectrometer through nanospray, and what you get is a spectrum. I'm showing the deconvoluted spectrum. You get a spectrum of the bacteriorhodopsin binding to the lipids, this PGP methyl ester lipid, and this is a glycolipid present here with this kind of stoichiometry, but it binds to the trimer. So bacteriorhodopsin trimerizes in the presence of the lipid. If we remove the lipid away from the protein, it falls apart into a monomer. So somehow these lipids then are important then for maintaining the stability of the assembly then of this complex. Without the lipids then, it just falls apart into the monomer. But you can make protein-lipid binding interactions then with this native mass spectrometry. Yes, question? Yeah. Right. Okay. So I didn't tell you the backstory. So the backstory is that bacteriorhodopsin, this particular sample, was used in the development of cryo-electron microscopy. So in the 70s, when people were thinking of cryo-electron microscopy, this was one of the first samples that they tried. And so when they eventually got some electron density that looked like the protein, they saw some extra density that they couldn't assign at the time. And so then this whole slew of researchers then went about to try to figure out if this was lipid or not. And so they determined that these two particular lipids were most likely present in the cryo-electron microscopy study. They also did X-ray crystallography to confirm that lipids were bound, but they couldn't figure out exactly what lipids were there. Then these other biochemical studies then, who focused on lipids then, they determined that these two were present, but they didn't know the stoichiometry. But they could sort of figure out from the X-ray crystallography density then where the lipids might be. We make the measurements then, we read the old papers, right? And we figured out, okay, these two lipids are the most prevalent ones there. And when we try to assign all the peaks, you can imagine what the raw spectrum looks like. There's peaks everywhere, right? But we tried to assign as many of these peaks as possible then, and these two kept coming up as the only two lipids that were present on the lipid. Now, there's some other stuff down here that we have, the minor stuff here, but these are the two main ones here. But, you know, we could go into a lot more work to figure out exactly what all these other peaks were. All right, so there's a lot of work left to be done on this one. But the point of this slide was just to show you that we can make protein-lipid interactions as possible. That's also possible with native mass spectrometry. Yeah. So then you can use it for identifying de novo interactions. Yeah, right. So if you just have a mass shift, you know that something that weighs 700 binds to your protein. All right, so something that weighs 700, if you think it's a metabolite, then you go to Aaron's talk this morning to figure out whether it's in the Metlin database, or you go to the lipidomics talk, which is downstairs, and figure out whether a mass of 700 matches to a particular lipid, or you resort to tandem mass spectrometry, what Josh was talking about, and try to fragment this 700 Dalton thing and see if you can get structural information. So there's a lot of detective work, I would say, to identify unknown ligands present. You just know the molecular weight, but then you have to then do some other experiments. God forbid you do NMR, but you don't want to do that. You know, try to stick with mass spectrometry. Okay, so this then segues then into top-down mass spectrometry. So let's say now I've identified a ligand binding to a protein. I think I know what the ligand is because that's what the medicinal chemist gave me, a drug. But now I say, yeah, there's a one-to-one interaction. I see a complex that a drug is bound to a protein. But can I get any additional information about where it binds to on the structure of the protein? Can I determine whether it binds to this position or this position or anywhere else? What can I do to do this experiment? Well, I'm going to resort to top-down mass spectrometry. I'm not going to give it to the NMR people. I'm going to try to see if I can figure out if I can do this by top-down mass spectrometry. So this is your intro then to top-down mass spectrometry. No enzymes. We just go straight from the mass spectrum of the protein to fragmentation. We're using the methods that Josh talked about earlier this morning. Same methods, either collisions, electrons, or photon-based dissociation methods. We produce fragments of the protein then, and these fragments then can be matched to a putative sequence. If we have the sequence of the enzyme of the protein, then we can try to figure out what these fragments are and how that maps out onto the sequence. We can get information then about the identification of the protein, but for our purposes, we can get information about binding sites of where ligands are present on a protein. Okay, so there's this kind of a fight going on between the bottom-up proteomics people and the top-down proteomics people. And yeah, 95% of the proteomics world uses bottom-up mass spectrometry, bottom-up proteomics, because the routine is already, the strategy is already sort of built, and it works well. Top-down mass spectrometry has some problems, but there are some unique advantages to top-down mass spectrometry. All right, so in bottom-up mass spectrometry, as Josh kind of alluded to this morning, then you take an enzyme, you digest it into peptides then, and each peptide is interrogated by tandem mass spectrometry to get the sequence. And then this sequence information then can be matched to a database to identify what that protein is. You'll get a lot more of this tomorrow, believe me. If the protein has a modification on the protein, you may or may not get that information because you may or may not recover that particular peptide when you do tandem mass spectrometry. With top-down mass spectrometry, we bypass the use of enzymes. We just go directly into fragmentation. And in principle, if a protein is modified, let's say this yellow symbol here, if we just cut the protein once and produce two pieces, then we should be able to localize generally then the location of that modification. And if we chop it up into smaller pieces, then we ought to be able to pinpoint then what residue this modification is present. So that's the justification. The other justification is that, let's say I have a collection of 3 proteins, it's the same protein, same sequence, but one form of the protein has one modification, the second form of the protein has this other modification, and this third protein has 3 different modifications. If I digested this sample into peptides, then yeah, I would get the location of all the modifications present on the polypeptide sequence, but I wouldn't know that I had 3 different forms of the protein with 3 different kinds of stoichiometries of the modifications. We call these proteoforms today. With top-down mass spectrometry, we're going to make the measurement of the intact protein first, so we know that there's 3 different forms, and then we can individually fragment them to identify the sites of the modifications. That's the justification. That's what we tell us, at least, why we're still working on this field. Okay, so the origins then of top-down mass spectrometry go back a long way. So around the time when electrospray ionization was first developed by Fenn's laboratory, I was at Pacific Northwest National Laboratory working with Dick Smith, and so we were just trying to see what we can do with electrospray ionization. And so we tried the experiment where we just took a protein and just busted it apart. We just used collisional activation, the method that Josh talked about, using collisions, broke the protein up into pieces then, and we tried to figure out then what the masses of these pieces were, and that matched the sequence. So we demonstrated that at least the protein could be fragmented into bigger pieces, and then those pieces then could be used then to identify the sequence of the protein. That was 1990. It wasn't until 1999 then when Fred McLafferty published this paper where he said top-down versus bottom-up protein characterization by tandem high-resolution mass spectrometry. He coined the term top-down mass spectrometry. In fact, this paper, I believe, also coined the term bottom-up mass spectrometry. So this paper is actually useful for both terms. But he kind of outlined then the utility of top-down mass spectrometry, where you can get sequence information directly by this method. So we weren't smart enough to come up with the top-down term, otherwise it would be in this paper. I trained with Fred, and so it's like that's fine if Fred came up with this term. And then later, then, as Josh also described, he came up with this other fragmentation method called electron capture dissociation, or ECD. And if you couple that with top-down mass spectrometry, now you get a lot of sequence coverage. You get a lot of sequence information because ECD combined with collisional processes is pretty much a very complete method then for getting sequence coverage, PTM coverage, et cetera, for proteins. So let me just remind you then, Josh showed this in his lecture this morning then, with CID, or collision-induced dissociation, or collisionally activated dissociation. It's the same method, just two different terms for the same method. That if you use CID processes, you break this bond between the carbonyl and the amide bond, and you get either a Y ion if it contains a C terminus, or a B ion that contains the N terminus. With ECD, you move one bond over. So with ECD, you break this nitrogen-carbon bond. You get a Z ion if it contains a C terminus, or a C ion if it retains the N terminus. Different cleavage points, but same utility. You get sequence coverage, and especially if you combine both methods. All right, so here's just a crude example. This is beta casein from milk, that same protein that Rachel showed in her lecture on ionization yesterday. Here's an electrospray spectrum of beta casein. It shows this A2, A1 isoforms that Rachel also talked about. And yes, this is since we're in Wisconsin, then we're going to study milk, and so there are two forms of casein, and you can measure this not only by MALDI but by electrospray ionization. And this particular group then also tried to sequence the protein by top-down mass spectrometry. I haven't read the paper fully, so I don't know if they actually found the sites of where the modifications are different in A1 versus A2. I'm sure it's in there. But you get these B ions, Y ions, because they use collisions then to fragment the protein to get sequence coverage. All right, so I want to do then combine then native mass spectrometry with top-down mass spectrometry, because I want to then try to figure out where the sites of ligand binding are, whether I can get some additional information on the structures of these large protein assemblies. And so you can study drug binding, metal binding, cofactor binding. If they bind to a protein and you can measure the mass of the complex, then you can further fragment to get some information about where they bind. But the method of how you're going to fragment the protein does make a difference. If you use CID, that collisional process, if this protein binds to this ligand, this green pill, and you subject that to fragmentation by CID, you're going to break the protein up into pieces, that's for sure, but you're also going to release that ligand. That ligand will just fly off because it's weakly bound in the gas phase or even in solution phase. And so the ligand will fall off, you'll just see a peak for the ligand, but you won't get any positional information if you fragment it this way. So these weak interactions are not stable to collisional activation. But instead, if you do that same experiment but use ECD, what you get are these peaks called hollow fragments. It chops the protein into pieces, but the ligand stays put to where it was originally bound. So you're going to get some fragment ions that retain the position of the ligand. And if you create enough of these hollow fragments, then you can map out the sites of ligand binding. ECD does not fragment non-covalent forces, interactions, and so those interactions should remain if you do this experiment properly. And so you can use this then also to map out sites of ligand binding. So our laboratory's been doing a lot of work then with drugs related to neurodegenerative diseases like Alzheimer's disease and Parkinson's disease. We studied the interactions of these ligand molecules to proteins like amyloid beta, alpha-synuclein, and we can map out positions of where these ligands bind. The one that we always study for a long time now is this compound called a tweezer, or CLEAR1. It binds to proteins like alpha-synuclein, and when it does that, it disrupts the aggregates that are formed in the brain. And so we're trying to figure out then where on the sequence then does this drug molecule then stick on alpha-synuclein, amyloid beta, and all these other neurodegenerative disease-related proteins. So here's an electrospray spectrum of alpha-synuclein. It binds to one molecule, this CLEAR1, represented by these red dots. It has a binding constant of about 2 micromolar measured by mass spectrometry, the same method I told you a little bit about earlier. And then when we fragment the protein plus this clear one molecule using ECD, then we can locate the binding sites to somewhere in the N-terminal region near one of these lysine residues near the N-terminus. And then we did that same experiment with amyloid beta. This is superoxide dismutase in Lou Gehrig's disease, tau protein, and tauopathies. And these gray regions then represents what our best guess is on where these molecules bind to in the sequence of these proteins. It always binds to a lysine residue because this molecule was designed to bind directly to lysine. And so it doesn't bind to every lysine residue, just to certain lysine residues in the polypeptide sequence. All right, so then how far can we go then with this technique then? If we have a larger protein and we blow up this larger assembly, then can we figure out then what these large pieces then we're going to get? Does that relate to anything related to the three-dimensional sequence or structure of the protein assembly? So in most cases, it does. And so if we use ECD, and we take a protein like this is alcohol dehydrogenase. It's a homotetrameric protein of about 147 kilodaltons in mass. And we fragment this protein using ECD. What this is called a fragmentation map. What we get are only N-terminal fragment ions. These are C ions coming from the N-terminus. And that's because if you look at the surface of this homotetramer complex, the N-termini of every subunit are on the surface of the protein. And so we're getting only surface residues then on the entire complex. And that happens for practically every complex we have ever studied, that most of the fragments that we get in this native top-down mass spectrometry using ECD are coming from the surface. So we can use this information potentially to solve unknown structures. We can tell you what's on the surface, what residues are on the surface of a complex by just doing this simple experiment, doing ECD native mass spectrometry. There are some other things that we discovered when we're trying to do these experiments. No, I've just told you about if I cleave a polypeptide sequence once, I should get either the N-terminal side or the C-terminal side. We call these terminal fragments, but in native top-down mass spectrometry in general, there's a lot of peaks in the raw spectra that are unassigned. And people just sort of ignored them for a long time. And so during the pandemic, we had nothing to do. And so we were just sort of sitting around looking for computational experiments to do. And so we had made the hypothesis that all the extra peaks might be what we would call internal fragment ions, meaning you break one bond here to make a terminal fragment ion, and then it further fragments into smaller pieces. Kind of like there was one example that Josh showed earlier today, where if you give it some more energy, it's going to further fragment into pieces. Well, when you have a protein, you have a big molecule, those pieces then could be called internal fragment ions. And maybe if we could assign these internal fragment ions, maybe that we can get better sequence coverage. Maybe we can cover the entire sequence if we can figure out how to assign these internal fragment ions. And maybe if a PTM is present somewhere in the interior of a protein, maybe we can find sites of PTM binding. But we need rules. We need super high resolution. We need super high mass accuracy so that we don't have any false positive readings in our assignments when we do these experiments. So during the pandemic, then we wrote a program called CLIPS MS. It's available on GitHub. Anybody can download it. And we've studied all the major forms of activation dissociation methods used in top-down mass spectrometry, UVPD, ECD, ETD, CID, etc. They all form internal fragment ions. And so we're trying to use this information then to develop rules on how to assign internal fragment ions and try to apply them for sequence coverage. So here's a raw spectrum, or this is the deconvolved mass spectrum. So this is in the mass domain. This is just CID of apomyelin, a single chain protein. And all of the peaks labeled with a star represent normal terminal B and Y fragment ions. So there's lots of peaks here that are unassigned. And so if we turn on our CLIPS MS program, then we can assign almost all of the peaks there. These are either internal fragment ions or neutral loss, meaning loss of water, loss of CO2, or loss of CO from an internal fragment ion. And so we can assign then most of the peaks now in a top-down mass spectrum. And this is quite useful then if we look at larger molecules. So lately we've been studying monoclonal antibodies, other proteins of therapeutic interest, mostly from Amgen. And so they've given us some antibody standards then to try. And if we fragment this by ECD and HCD, that CID method that Thermo calls CID in an Orbitrap instrument, we get, with just raw ECD, we get about 65% sequence coverage. So we can cover 65% of the sequence by ECD. A lot of the fragment ions we assigned are internal fragment ions that contain glycosylation sites. If we just do HCD, we get about the same sequence coverage, but only if we turned on internal fragment ions. If we ignored internal fragment ions, we'd only get about 27% sequence coverage. And then if you combine the two data sets, then we get about 75% sequence coverage using ECD and HCD. The last paper we published just earlier this year then does middle down mass spectrometry. So if you're hip to these different techniques that top-down people use, if you cut the antibody here at the hinge region using this fabricator enzyme, now you look at the 20 kilodalton pieces, you get about 99% sequence coverage then just by using ECD. So cutting the protein down into smaller pieces helps, but you can get quite a large amount of sequence just directly from this 150 kilodalton protein. All right, so I'm going to end here just by telling you that I didn't cover all the other dissociation methods for top-down mass spectrometry and native top-down mass spectrometry. All of these other methods like UVPD, SID, I covered a little bit about CID and ECD. These are all methods that are useful. Not any one method will fit all buckets for your goals. So it depends on what the goal of your experiment is as far as how you do this experiment. And I didn't talk about online chromatography. You see, that's kind of missing, right? Online chromatography with native mass spectrometry is still missing, especially with native top-down mass spectrometry, other than SEC, which is kind of a terrible resolution technique for chromatography. But I just pull up this paper from Ying Gu's laboratory from Wisconsin. They just published this paper on something called mixed bed ion exchange chromatography. And this is compatible with native mass spectrometry. So I invite you to look at this paper that was just published in Journal Proteome Research. Using this mixed bed ion exchange chromatography column, then they can resolve different proteins, native proteins, and then do online, then top-down mass spectrometry using this technique. Here's one example. This is a human heart extract. That's all they ever study is human hearts, or pig hearts, I guess. And you get all these different complexes present under this really ugly-looking chromatogram. But if you pull out the EICs for each of the individual proteins, this is malate dehydrogenase. They can fragment this protein to identify what this protein is. This column is not commercially available, as I checked the other day. Is anybody from Ying Gu's lab here? No. Okay, I'm going to tackle Ying if I see her later this week, because this is a column that was made by LC Packings with Annie Alpert's company, whatever that company is. So it's not a catalog number yet, but I was hoping to get a column from Ying's lab to try this. So if you want to do chromatography with native mass spectrometry, this might be the way to go. All right. So with that, I hope I've just an introduction for native mass spectrometry and top-down mass spectrometry. For us, it's useful for structural biology, but for you, it might be useful for some other technique. So thanks very much, and I'd be happy to answer any questions. Questions? Yes?
So I was really interested when you showed the antibody that you cut and were able to increase the sequence coverage. So I'm not 100% clear why that makes such a big difference to be able to, you're basically cutting off the epitope.
No, the epitope. Oh well, there's no epitope. I mean, there's no epitope bound to this. So it's just the antibody. Um, but if I cut the protein using an enzyme, so we call that middle down mass spectrometry, just because we're making big pieces. So we cut it here, and so this piece, and this piece, and comes into 3 pieces now, and they're about 25 kilodaltons. It's a little bit smaller that way. It's a little bit easier to handle, a little bit easier to fragment then.
So that makes, okay, that's what I'm understanding.
Yeah.
The fragmentation easier because you have smaller pieces.
That's right. That's right. The nice thing about ECD, though, is what I didn't say is that there's 16 disulfide bonds in this protein, and ECD will cleave those disulfide bonds directly, and so that's nice to combine that with ECD. Yes.
Is there any limitation to what kind of interactions these methods may miss?
Yeah, so the question is, are there any limitations to the type of interactions that native mass spectrometry maybe wouldn't address? For the longest time, I thought that hydrophobic interactions would be completely transparent because hydrophobic interactions require water. And I'm putting this protein in the gas phase with no water. So I've evaporated all the water. And so how does a lipid bind to a protein? Usually it's through the tails that form hydrophobic interactions to some parts of the residue. And so if the water's missing, then how is that even stable? Well, some of these lipids have charged polar groups. And so it could be that the charged polar groups are allowing it to stick to the gas phase protein so that I can still measure that interaction. We've not studied that yet, and so I would guess that completely hydrophobic molecules sticking to a protein would not be stable under native mass spectrometry. I would predict that, but no one's ever published that yet.
But even things like hydrogen bonding and really weak interactions are captured in other.
So we can measure, we've measured interactions at millimolar KDs. Because there is a stable electrostatic interaction that still holds it together. So the KDs in the gas phase are not the same as the KDs in solution. In fact, in the gas phase, there's no equilibrium. There's no off rate. It's just a straight affinity. But in solution, we're measuring an off rate, on rate, right? So it's a little bit different, which is one reason why when I was at Pfizer, I would never tell the medicinal chemist that I can just rank order the binding of all the drug molecules you're making just by the gas phase measurement, because I know that different gas phase stabilities will be reflected in our measurements compared to what the medicinal chemists would care about, which is KD or IC50s or something like that. Yep.
Is native MS possible?
If you read the early papers of MALDI, especially by Franz Hillenkamp, they were showing some of the same enzymes proteins that I was showing, and they were showing aggregates or the right sizes of some of these processes. And so I normally would have thought that the matrices that Rachel showed that were useful in MALDI, they're very acidic. And so I would think that would have disrupted the interactions. Plus, when the laser hits it, there's a lot of energy going on. I would think that would disrupt it. And so they were, well, that's for UV, right? And so later, if you follow the progression of MALDI, there was a group of people that was saying either IR lasers would be better. There was a group of people that was trying to promote the use of ice as a matrix for IR MALDI to look at large protein complexes. And then there was this phenomenon called single shot MALDI, where it's like you fire it once and you magically get a spectrum of a native assembly. That has kind of died off right now just because it was very hard to reproduce. There's something still intriguing in there fundamentally that they're getting sizes of complexes this way. but it never caught on, as MALDI didn't catch on. Well, MALDI's kind of almost dead, other than imaging and some of these other methods. So if anybody want to get some of the back into MALDI, that would be a method then to look at.
I have a question on this one. What's the goal for you and Amgen on this? Is it to know they made the right sequence, or is it to like map oxidations and deaminations, or all of that?
Well, so the latter one. So the one of the goals is to in pharmaceutical companies, they want to do these stress conditions. They want to take their therapeutic protein and stress it under high temperature, high oxidation states, because they want to be able to produce a therapeutic drug that if a doctor in Africa puts it on the windowsill of a counter and stores it there for 3 weeks, it's still going to be stable. And so they want to understand then where are possible modification sites on the antibody so they can mutate those sites out. Another thing that I didn't show on this slide is that all of these companies are making antibody drug conjugates. So they're covalently labeling sites of small molecule drugs attached to the antibody. And so they would love to then map out the sites of the antibody, the drug bound to the covalent drug bound to the protein. So if we can say that if we can just do one experiment, get 100% sequence coverage, we could just tell them exactly where the binding sites of the drug molecules are and what's the stoichiometry at each site. Anything else? Yep.
For the internal fragments, have you guys, or has anyone in the field done a lot of MS3 experiments at this stage to kind of chase down where you find
Not in a. So the question is, has anybody looked at MS3 to confirm or disprove internal fragment ions? And the laboratory at Michigan, now she's at Florida State, Kiki Hawkinson, they proposed to do MS3 experiments to confirm these internal fragment ions. Remember, internal fragment ions are, let me go back. Too many bullet points. These things here, these masses overlap with a lot of other possible assignments. And so if you did MS of these, be an MS3 experiment, then you can confirm what the sequences of these internal fragment ions are. So the Hawkinson laboratory proposed to do that. They showed a few examples at an ASMS talk last year and this year also. And we are on a paper with them to propose to do MS3 experiments on these. It's especially problematic for electron-based fragmentation, that you get all kinds of different assignments because of +1, -1, protons flying around, radical sites going on. And UVPD is just another can of worms that I don't even want to touch. And so if we can figure out if these are real or not, and how rigorous do we, what kind of rules do we need to rigorously assign these, then that would advance the field. But yes, but it's not easy to do MS3 on these experiments. Yep. Ultraviolet photodissociation. So using a laser 193 nanometers or 213 in the commercial thermal instruments. That produces every letter in the alphabet type of fragment ion for proteins. So it just blows up the protein into all kinds of pieces then, and it's quite effective for fragmenting any kind of molecule. Yep.
Thank you for the for weak protein-protein interaction, when do some kind of cross-linking before subject to
You could do chemical cross-linking of whether protein or other ligand to your protein or other biomolecule, and then subject that to tandem mass spectrometry. That is possible, but it's experimentally a little bit problematic because it's really hard to get one cross-link from the ligand to the protein. What you're going to get is like many cross-links all over the place, and then sorting out the right peak that represents the one cross-link you're interested in is a little bit challenging. But people do cross-linking with bottom-up mass spectrometry to get interaction maps that way.